Hi. Following the discussion on basic genetics, we will go through some important aspects of basic embryology. So, embryology is the process of embryo development. It starts with the fertilization process where the sperm and ovum get together. As we discussed under genetic discussion, the sperm and ovum have a haploid karyotype, and so when they mix together, they form the diploid pattern, and there is genetic exchange of information from both the paternal and the maternal side. That's why the baby has features from both parents, and actually they may have features from family members in the parents' family. They may skip generations even because the genes are present transferred from the families, but the representation or manifestation depends on the epigenetics. The zygote formation, the stages include cell multiplication. Initially, it multiplies to multicellular form, and then the organogenesis progresses from the fourth week onwards. It continues to mature, and the development and maturation of the organs once they are formed takes place. There are huge changes. I will show a couple of illustrations. The uterus holds the zygote. The wall of the uterus allows the zygote to implant in it, and once the embryo is implanted, the placenta attaches to the uterine wall, and the yolk sac is attached to it as well. So the cord connects the placenta to the developing fetus and acts as the supply of nutrition. The various aspects that affect the placentation or the umbilical cord related problems can affect the baby as well. So you can see here the different stages. So the zygote is formed, the unicellular stage after the fusion of the sperm and the ovum. So the sperm enters the ovum, and then it's a fertilized ovum or a zygote. It divides first into the diploid stage, the bicellular form, multicellular form. After a few multiplications, it's called the morula. And after the morula, there is a yolk sac formation, the blastocyst and the blastomere. So the central space is the primary yolk sac cavity, and we have the main the groups of cells which lead to different forms of the body, different areas of the body. So the inner lining forms the endoderm, the middle lining becomes mesoderm, and the external lining becomes the ectoderm. So we have parts of the body derived from each aspect of these. So the gut, the lungs and so on develop from the endoderm. The mesenchyme leads to the muscle skeletal system, and the ectoderm leads to the skin, and the neural system develops from the neuroectoderm. So this is an image based version of it. So fertilized egg, 2 cell stage, 4 cell stage, 16 cell stage, the morula, the blastocyst, and then you start seeing the fetus developing. By 4 weeks you start the organogenesis process. The beating heart can be seen on the scan as early as 4 to 5 weeks. The fetus of 10 weeks has limbs, and you can see the eye buds. The head has started enlarging, the neuroectoderm has folded, and around 16 weeks onwards you have the relatively fetus looking, baby looking fetus, though very immature. And from 20 weeks you can see the full structure as we would see normally, even with the digits. And after 24 weeks eyelids start opening gradually. We start saving babies from 23 to 24 weeks onwards, and these babies are born very fragile, immature skin, very thin transparent skin. They need the highest level of intensive care to help them survive. So this is how the fetus lies within the placental cavity, the amniotic cavity, and this is the placenta and the umbilical cord connecting. So more defined images reconstructed, and you can see how the definitions start appearing. So the ear bud, the nose bud, and then the shape of the ears and nose start forming. And you can see the umbilical cord is very bulky because the yolk sac, the intestinal rotation happens around 12 to 14 weeks, and then it goes back in. So initial stages you have the umbilicus, which is bulky because the intestines are external and then they go back and rotate. So that is anomalies in this rotation. A return and rotation problem can lead to malrotation of the intestines, which can cause intestinal obstruction and is a serious emergency in babies. So these are the first trimester changes. So the pregnancy is broken up into 3-month segments. So the first 3 months is the first trimester, when most of the organogenesis takes place. The second trimester is the maturation of the organs once they are formed and the growth of the fetus starts. The third trimester is when the majority of the growth happens, and there are some important changes happening as well, like the antibody transfer from the mother to the baby, which is a passive immunity, mostly happens in the third trimester. The size of the fetus nearly doubles during this stage as well. So from 8 to 12 weeks is the start of the organogenesis. There is a high risk of teratogenic influences during this period, so radiation, medication, etc. One of the important aspects that we should know is folic acid taken peri-conception, that is before the conception and continued during the early stages of the pregnancy, is known to reduce neural tube defects, and it may also reduce other birth defects. This is because of the rapid multiplication of the cells and the importance of folic acid for RNA and nucleic acid synthesis. We have abnormalities, and these can be malformations where the structure itself is not formed normally. It can be deformation where external forces kind of compress. So it can be multiple pregnancy where one baby's foot is pressing on the cheeks of another baby, so that can cause deformation. A baby may have reduced fluid within the amniotic cavity, that is oligohydramnios, and when the baby is developing in that kind of setup, they may have features which are abnormal. A baby who has a cleft palate may have an associated large tongue, small chin, so this is a deformation sequence related to the underlying problem. Disruption happens when there is blood supply interruption, or it may be a floating amniotic band. The amniotic band lines the amniotic fluid cavity, and when the amniotic band portion of it separates, it can wrap around certain portions of the baby's limb or sometimes even the genitals, and it can result in amputation of that portion. So these kind of changes are called disruption. So teratogenesis would contribute to malformation. One example of teratogenesis is the thalidomide tragedy, when thalidomide was used for morning sickness in the late 50s to 60s, and it was noted that was a huge rise in babies with phocomelia, where the proximal segment of the limbs do not develop. So this was later attributed to the thalidomide, and it's a huge public health issue. Teratogens have to be identified, and pregnancy, there should be warning clearly mentioned that there shouldn't be anyone planning a pregnancy should not take. So it's a planning stage, because you may not even realize you're pregnant in the early stages, because organogenesis takes place in the very early stages of pregnancy. And so if you're exposed, so as soon as you start planning conception, take folic acid and avoid exposure to any medication that could be teratogenic. I explained about the sequence. So one example is Potter sequence, where the primary problem is an abnormal fetal kidney. So because the kidney doesn't produce enough urine, there is a low amniotic fluid level. When there is a low amniotic fluid level, the baby's lungs don't develop well, because the chest doesn't have the room to move when there is less fluid around it. The baby's face looks abnormal in these cases as well. So if the baby has bilateral kidney problems, and the kidney is not working, you may lose the baby, because the baby cannot breathe after the delivery. So this kind of problem is called Potter sequence. This is how a baby with Potter sequence looks like, and because the baby is squashed, the amniotic fluid allows the baby to float, and the limbs are kicking and moving. So if that is not there, the limbs are squashed, and you can see how they are bent. And you have the facial appearance with low set ear, beak, nose, epicanthic fold, the eyes are pressed against as well. And there is lung hypoplasia, so the chest is small, and the babies, we find it very difficult to oxygenate them. The lungs will rupture when we try to give pressure ventilation to keep the lungs open. And bilateral renal agenesis is the start of the problem, reduced urine output and oligohydramnios. So next we will be looking at basic biochemistry, and this is just a simplified form of embryogenesis. Obviously embryogenesis is very complicated. Embryology of each organ system is very complicated, but in the basic level it suffices to know the different stages of development, the role of teratogens, what is the different forms by which the fetus can be malformed. I said malformation, deformation and disruption. So if you remember these, that should be enough. Thank you.